Here now the comes the what is then the role of the lymphocytes. Here the lymphocytes are the key cells of the immune systems, and they are formed from the stem cells in the bone marrow, whereas the differentiated into the B and the T lymphocytes, in the B lymphocyte in the bone marrow and T lymphocyte at the thymus. Here the B lymphocytes are processed in the bone marrow, and the B lymphocyte synthesize the antibodies that provide the humoral immunity. Whereas the T lymphocytes, the T cells, are processed in the thymus, and the thymus and provide the cell-mediated immunity. And both the B and the T lymphocytes are capable of recognizing the antigens. Given B or the T cells can recognize only one particular antigens. Then the main part come here is the humoral immunity. Here these operate against the extracellular antigens and through the antibodies. And these antibodies are the plasma proteins belonging to the gamma globulin fractions, and they are also known as the immunoglobulins. Basic unit of immunoglobulins molecule consists of the two parts. There is the heavy chain and the other is the light chain. Heavy chain are joined to each other by the disulfide bond, and the one light chain is joined to one heavy chain by the disulfide bond. Here we have the structure of this immunoglobulin. In this, there is the N terminal and there is the C terminal. Here it is the light chain and this is the heavy chain part. And in this, these are the variable regions and these are the constant regions. And in this, there is a disulfide bond. And these are the hinge regions. And this is the structure of the immunoglobulin. Then the immunoglobulin light chain. These light chains are smaller than the heavy chains and their molecular weight is about 23,000. And they are of the mainly of the two types, the kappa and the lambda parts. Here, but only the kappa or the lambda, or one of them can be present in the one immunoglobulin. One immunoglobulin has only one type of the light chain, either this kappa or a lambda. The C-terminal half has a constant amino acid sequence and this is called the constant region, whereas the N-terminal has a variable amino acid sequence, so this region is called the variable region. And the immunology, here the immunoglobulin heavy chains have a molecular weight of higher than those of the light chain, so they are called heavier one. They are around the 50,000 to 70,000 daltons, and they are of the five types: alpha, delta, epsilon, gamma, and the mu. Here, the one immunoglobulin has only one type of heavy chain. In one type of the heavy chain, C-terminal part has the 3/4 has the constant amino acid sequence, whereas the N-terminal has 1/4 and has the 1/4 has a variable amino acid sequence. And the C-terminal has the 3/4 of the heavy chain is known as its constant region, whereas the N-terminal 1/4 of the heavy chain is known as its variable region. Here we can see these are the variable regions and these are the constant regions denoted by V and denoted by C. The light chain and heavy chain are folded to form some globular domains, and the variable region of light and heavy chain come together to form antigen binding sites. This is the ribbon-shaped structure of this immunoglobulin. The variable region of the light and the heavy chain have the three hypervariable HV regions, and the amino acid sequence is extremely variable in this hypervariable region. And the HV, this hypervariable region, constitute the antigen binding sites. And these HV regions are also called the complementarity determining region or the CDRs. They determine the complementarity between the antigens and the antibodies. Here, these are the hypervariable regions determining regions called the CDRs, and these are the hinge part, and these are the disulfide bond, and this is the structure of the immunoglobulin. And only a small region of antigens is known as the epitope. Earlier we have discussed that only a small portion of antigen can be called as the epitope, and it is recognized by that of the antibody. Here we can see with the diagram, here the antigen is recognizing this epitope part from its variable region side. The region of antibody that contracts the epitope on the region of the antibody that contacts, okay, this epitope is contacting with these antibodies through the paratopes. These are called the paratopes and these are called the epitopes. Then the some of the experiment was done, and to check the activity of the to check the structure of the immunoglobulin, and they found at when the action of papain was done, this is the proteolytic enzyme. This papain cleaves the antibody molecules, and the molecule is broken up into two identical FAB fragments and one FC fragments, and each FAB fragments can bind antigen, and the fragment antigen FAB means the fragment antigen binding, and FC means the fragment crystallizable, and the FC fragments can be easily crystallized, and the papain treatment has thus shown the function of the different region of the antibodies. Here, diagrammatically shown that the action of the papain, the antigen binding sites, and the these when undergoes the papain cleavage, the two fragments are generated, mainly the FAP fragments and the FC fragment.